to Science Fantastic with Professor Michio Kaku. On Science Fantastic every week, we profile some of the most amazing developments in science and technology, science that touches your life. Today, we're going to talk about the eternal question, what is life? Back in 1944, quantum physicist Erwin Schrödinger wrote a book, What is Life? Back then, there was a theory called vitalism. And of course, there's no vitalism, there's no spirit, there's no ghost out there that animates object. But that was a dominant theory back then. Just like when Einstein battled against the ether theory, Einstein banished the ether theory to create relativity. Well, Schrödinger wanted to banish this vitalism theory to replace it with the quantum theory of molecules. And he made a conjecture for the ages. He conjectured that there should be a molecule, an unnamed molecule of some sort that had a code on it. And this code was replicated across generations, and that was the basis of life. Well, the book inspired Francis Crick and James Watson to find that master molecule, which we now know as DNA. Well, again, with us today is Sir Paul Nurse, winner of the Nobel Prize in 2001 for physiology or medicine. His book is called What is Life? 5 Great Ideas in Biology. We should point out that he's a former president of Rockefeller University, current director of the Francis Crick Institute in London, and now author of this exciting book that I highly recommend, What is Life? 5 Great Ideas in Biology. So, Paul, glad you could be on Science Fantastic.
Well, I'm pleased to be speaking to you, Michio. I'm not sure I'm going to be offering you a theory of everything, but I'm hoping to offer some theories about biology that help us understand life.
Okay, well, that's good enough for me. The first question that we always ask our guests is, when you were a child, think back to when you were a young kid. What inspired you to get interested in science, and then specifically biology and later genetics?
Well, I think my first excitement was looking at the stars. I lived in London, and it wasn't always that dark, but I saw the stars, I saw planets going, changing their positions during the different parts of the year. And in particular, I remember when I was 7 or 8 years of age of seeing Sputnik 2. Sputnik 2 was the second satellite that was sent up by the Russians, had a dog in it called Laika, and I read in the newspaper it would be crossing over London at a certain time in the evening. I was in my pajamas watching for it from the front garden, and there it was, this star moving across the sky. I remember chasing it down the street, following it and telling everybody, look at that, it's a satellite, like the dog's up there. And I think that was the start of my interest in science.
And then what steered you in the direction of biology? I understand you had a romance with butterflies and beetles.
Yes, I used to have a long walk to school, and I went through several parks, and I got really quite interested in the changes in the seasons. Obviously, leaves growing and then falling off in autumn and so on, and just watching the changes in natural life. And I noticed the butterflies, I noticed beetles, I noticed birds, and so I got interested in natural history as well. So it was a combination of astronomy and natural history.
Okay, now your book is divided into 5 great ideas in biology, but you also try to summarize the 3 main features of life. Now, the definition of life is quite complicated, but you mentioned 3 distinct features common for the life forms that we've seen, and we maybe see this in outer space when we encounter alien life one day. So what are the 3 basic principles that you outline?
Yes, so you're right. I pull on 5 great ideas. Maybe we'll talk a bit more about them later, which are well-established ideas, ones perhaps that us biologists don't talk about enough. And then I pulled those together and defined these 3 principles. And they are, well, the first is that living things evolve. and they evolve by a process of natural selection. This is, of course, Charles Darwin's great idea. And to be able to do that, a living thing has to reproduce, of course. It has to have a heredity system determining how they work, and that's the DNA you were just talking about. And thirdly and critically, that heredity system must exhibit variability, and upon that variability, selection can act. If you put those three things together, then you develop organisms that can evolve, that can adapt to their environment, adapt to their way of life in the most effective way. So they end up looking as if they're designed because they are fit for purpose. They haven't been designed, they've just evolved by natural selection.
Welcome back to Science Fantastic with Professor Michio Kaku. We're very delighted to have with us Sir Paul Nurse, winner of the Nobel Prize in 2001 for his pioneering work in physiology or medicine. And the book goes to the very heart of all of biology and medicine. What is life? 5 great ideas in biology. So let's talk about the first great idea, which is the cell. Now, years ago during the Middle Ages, we had this theory called vitalism. There was this strange spiritual ghost-like thing that pervaded inanimate objects, and boom, suddenly they became animate. But just like Galileo introduced the telescope, which opened up our eyes to the heavens, Robert Hooke popularized the microscope. So tell us a little bit about the impact that the microscope had in our view of life.
Well, the basic unit of life, it's really life's atom, I think of it, is the cell. The cell is the simplest entity that exhibits the characteristics of life, and all living things, at least as we know them on Earth, are either a single cell or a collection of cells. And of course, we are examples of that. We're made up of trillions of cells. And these were first observed by Robert Hooke, working in Oxford, and he turned a microscope, which had been invented 30, 40 years before in the Netherlands, and turned it to living things. And he took a slice of a piece of a plant, put it under the microscope, and there he saw cells, little boxes all in a row, and it was the first observation of the basis of life, the cell.
And didn't he also draw a picture of a flea so that living organisms could also be shown in all their full glory?
Yeah, there's a great picture of a flea. It's actually, I've seen this picture. It's at the Royal Society in London, where I was once president. And I have on my sofa at home a cushion with a picture of exactly that flea all over it.
So I guess in the same way that Galileo really shook up our understanding of the universe, Hooke and his microscope really shook up our understanding of life, that life occurs in cells. There's no vitalism that animates inanimate objects, right?
Absolutely did. And just a little after him, Leeuwenhoek, who was working in Delft in Holland, took a scraping from between his teeth, this was only 10, 20 years later, and put it under a very simple microscope and discovered bacteria. In other words, discovered that some life forms only were made up of a single cell. He was a bit upset about it, though, because he was rather proud of his dental hygiene, and I don't think he liked the idea of all these little microbes crawling around between his teeth.
And didn't this have a huge impact in medicine? Because all of a sudden we realize that diseases are also spread by these cells, these bacterial cells.
Well, it had a big impact in all sorts of ways. First of all, infectious diseases are often caused by bacteria, of course also by viruses, which are even smaller than bacteria and only are properly alive when they're in other cells. But there was a German pathologist called Rudolf Virchow who identified the fact that all cells come from previously existing cells, and he had this idea in the 19th century that the basis of all disease, it was to be found in the malfunctioning of human cells. Something was wrong with the cells. That would cause a problem with the tissues and organs, and you would get disease. And it's a very fundamental discovery, as you rightly said. that cells are critical for disease, both in infections and all other diseases which are defects as a consequence of damage or something else influencing the functioning of cells.
And the next great concept you mentioned is the gene. And here we have this monk, Mendel, who actually wrote down the foundations for genetics, but he was pretty much ignored during his lifetime, right?
He was. He's a favorite of mine. I call him the gardening monk. In the middle of the Cold War, I went to his monastery, which is in Brno, now in the Czech Republic. And I visited his garden, and he had a large garden. He had a large greenhouse there. It was really like a research institute in the monastery. And he crossed lots and lots of different pea plants together with different characteristics, different colored flowers, different types of seeds, tall plants, short plants. And when he crossed them together and counted the progeny that they were produced, and he was trained as a physicist initially, so he was sort of used to counting things, he found that these different characteristics separated out in very simple ratios like 3 to 1 and ratios like that. And from this data, he concluded that the inheritance of characteristics was all related to particles, indivisible particles, which turned out to be genes, being inherited from parent to offspring, and that was the basis of heredity, and of course was explained by the code script that Schrödinger predicted, and which turned out to be DNA.
And we mentioned Francis Crick and James Watson, who did this pioneering work, writing the paper for the ages, deciphering the DNA molecule, which had the code of life. Can you explain to me the sea change that took place? This was historic, starting from a position of being blind to a position where we could actually unravel the code of life. Could you elaborate?
This was a great discovery. It actually started in London with Maurice Wilkins and Rosalind Franklin, who produced X-ray crystallography. This is a method which allows you to look at the structure of molecules. And they produced beautiful images. They didn't interpret them. It was Watson and Crick who interpreted them and determined that it was a double helical structure. And the way to think of this double helical structure is like a ladder with rungs going across it. And each rung consists of two bases which pair with each other and with no other bases. So there is a base A that pairs with a base T and a base G that pairs with a base C. And so you could produce an exact complementary copy on one rung of the ladder compared with the other. And if you rip these apart, these could form a template for an exact copying of that gene. A fantastic discovery, 1953, and has really led to the birth of molecular biology and molecular genetics.
And there's a famous picture of Watson and Crick next to this Tinker Toy, this Lego model of the DNA, this double helix. And if you look carefully at that picture, you can see that it's composed of individual atoms joined together whose bonding angle was determined by quantum mechanics. And so using the bonding angles from quantum mechanics, you could then assemble atom for atom this double helix. I mean, that is mind-boggling, right? Atom for atom, being able to construct a molecule that carries the code of life itself. Incredible, right?
Absolutely incredible. Exactly as you say, you move from the quantum mechanics of physics to the bonds of chemistry to the hereditary material of biology. And
this culminated into the Human Genome Project. I mean, think about it, a $3 billion project to decode life itself. And now we can decode the secret of life itself just for a few $100s. Isn't that amazing?
It is amazing. I remember my laboratory was involved in sequencing the third eukaryote. That's the same type of organism as a human. We did it in about 2000, a couple of years before the human genome, and it took us 120 people 3 years to do. And today we can do it in less than 1 hour on a machine.
Okay, now speaking about genetics, you tell a rather interesting story, a rather personal story. I was kind of surprised to read it. When you found out getting your birth certificate that your parents, the people that you thought were your parents who raised you as a kid, were actually your grandparents, and that your sister was actually your mother. Wasn't that a shock from the world of genetics?
It was, and I'm a geneticist, and my own genetics have been kept secret from me for over 50 years. And in fact, I only discovered it by accident when I was applying for a green card when I was working at Rockefeller University in New York. And I was rejected by Home Security, who didn't like my birth certificate, that didn't name my parents. Said I was British, but didn't name my parents.
Welcome back to Science Fantastic with Professor Michio Kaku. Today we're talking about what is life with Sir Paul Nurse, winner of the Nobel Prize in Physiology or Medicine. It's a great book. The subtitle is 5 Great Ideas in Biology. Well, we left off, the gene is the second of these 5 great ideas in biology, and you had the shock of a lifetime when you got your birth certificate and it listed for your parents, your grandparents. So the people who raised you were your grandparents, and you found out that your sister was actually your mother. Wasn't that a shock coming from the world? Yeah?
It was a shock, and I was 55, 56 years of age, so it was later in my life. When I looked at the birth certificate, the name of my mother was my sister. And then I looked to see the name of my father, and there was just a line, no name. And so what had happened is that my mother had got pregnant at 17. She was sent away to her aunt, 100 miles away from London, gave birth to me, and my grandmother came up and, to protect her daughter, pretended to be my mother, took me back, and my grandparents brought me up. And I was never told. And in fact, when I did find out, both my grandparents who brought me up and, unfortunately, my mother had all died. So I couldn't even talk to them about it, and nor do I have the slightest idea who my father is.
Now, today we have Ancestry.com, 23andMe.com, where you can easily find out your family tree going back hundreds of years. So have you ever done that to try to find out who your father really is? Perhaps
yes.
His genome is also registered, yeah?
Yes, I have done that because exactly as you say. And I have discovered 2 people who have a related DNA sequences at the level of a cousin, so not that distant. And I did try to contact them through the sites that these sequencing companies have, but they've never got back to me. So they are probably my relatives, and we share a common ancestor who will be my father, but I haven't managed to take it any further.
Okay. Well, let's go to number 3 on your list, which is the Darwinian theory of evolution, which we'll talk about after the break. And we'll talk about a gene called CDC2 that you pioneered. Welcome back to Science Fantastic with Professor Michio Kaku. With us today is Nobel laureate Paul Nurse, winner of the Nobel Prize in 2001 for Physiology or Medicine. The book is called What is Life? 5 Great Ideas in Biology. Well, let's talk about the third great idea, which is evolution, which of course was perfected and promoted by Charles Darwin. And Darwin wrote a famous tree in his notes, a tree of life, where at the trunk there was one life form that gave birth to branches with all the other life forms. If that's true, that means that even the yeast cell, the lowly yeast cell, is our cousin, separated by hundreds of millions of years. And therefore the genes of yeast must have a counterpart in our genes. And I understand that you pioneered the work with a gene called CDC2. Explain to us why the gene CDC2 is important for medicine, for physiology, and for understanding Darwin's theory of evolution.
Yes, I'm delighted to tell you about that, because what my research has focused on is what controls the reproduction of cells from 1 to 2. We've already discussed that the cell is the basic unit of life, and therefore the basis of growth and development of all life is cell reproduction. And I was interested in finding out how that is controlled, and discovered, working on the simple yeast, as you clearly have just explained, the simple yeast, I discovered a gene that controlled that reproduction. It acts really like the accelerator in a car that determines the speed at which the cell will reproduce. When it's activated, the cell begins all the processes that lead to cell reproduction and then completes that process and divides into 2. So it's a master regulator of cell reproduction. Now, that's in yeast, but at the time when I discovered it, which was in the 1980s, nobody really thought that such a simple cell reproductive process as you find in yeast could also be controlled by the same gene in a more complicated cell, our own human cells. So to test that, what I did is I took a gene library of human cells, sprinkled that library onto yeast cells that were defective in this gene, which was called CDC2, and looked for yeast cells that now could grow and reproduce because they picked up a gene from the human gene library that could compensate for a defective CDC2 gene in yeast. And amazingly, that experiment worked. And this was long before we had the sequence of either yeast or humans. And when I and my colleagues recovered that gene back out of the yeast and sequenced it, and it took over a year to do it, or at least some months to do it, we discovered the human gene. And this was amazing, because what it meant was that the controls over cell reproduction have been the same for 1,500 million years, 1.5 billion years. So really, at the origins of this type of eukaryotic life form, the control over cell division was set in stone, and it's remained unchanged. So yeast cells really are our cousins. They're very distant cousins over 1.5 billion years ago, but they are our cousins. And that's very important, because it means every living thing we look at, and I'm looking in my office here at a plant, for example, will be controlled by the same process. So it shows that the tree of life, as Darwin drew it, is connecting all these different types of organism, and all the cells in them are controlled in the same way through the CDC2 gene. This just blew my mind, as it's sure you can imagine.
Now, Charles Darwin, of course, got into big trouble because back then the church said that the universe of life was designed. But in your book, you say that life does have a purpose, not the purpose necessarily that Charles Darwin came up against in the 1800s, but there is a purpose to all this. So what is that purpose coming from genetics?
Yes, and I use the word purpose in a particular way. It's not purpose as we humans normally think about. We act with purpose. What it means really is that living things behave in a coherent, connected way.
Welcome back to Science Fantastic with Professor Michio Kaku. Once again with us on the line today is Sir Paul Nurse, winner of the 2001 Nobel Prize in Physiology or Medicine. The book he's written is called What is Life? 5 Great Ideas in Biology. And when we last left off, we were talking about the fact that a gene that he discovered in yeast cells had a commonality with our own cells, even though that goes back billions of years into the past. So I guess when Mother Nature finds a chemical reaction that is good and suitable, Mother Nature capitalizes on it and takes variations of it. So again, that gene is called CDC2. So, Paul, hasn't that really changed our attitude that we are in some sense part of this tree of life, that the genes that we have, some of them go back billions of years?
Yes, it has, and I should say that it wasn't just my work. It was also work by Lee Hartwell, who was working in Seattle at the time, working on a different yeast, and he discovered this gene as well, it's very similar to the gene that I got, and we showed it was the same in humans and a number of other living things in between. So it really does demonstrate how highly conserved all these different organisms are in controlling their cell reproduction. It's a highly conserved process and really confirms Charles Darwin's tree of life.
And you also mentioned a purpose, not necessarily a human purpose, but there is a guiding principle behind all of this, right?
Well, there is, because, you see, I was explaining, living things behave as if they have purpose. They grow, they maintain themselves, they reproduce, they adapt themselves perfectly or as well or very well to their environment and to their lifestyles. And these are all sort of purposeful behaviors. And coming about by evolution by natural selection, which allows these different life forms to become better adapted to the environment in which they find themselves. And that is a key way in which you apparently get design, but without a designer. And it's a very simple, but very, very important and clever idea.
Now, also in your book, you mentioned life as information. Now, usually we don't think of information and life having much in common. One is based on digital zeros and ones on computer tape. But DNA, in some sense, is like computer tape. 4 nucleic acids, A, T, C, G. Could you elaborate on that?
Yes, I can, and it's very important, really, because what I said is that living things are chemical machines, but they're also informational machines, because the chemistry makes sense in terms of biology when you think about it in terms of managing information. And the archetypal example is, as you mentioned, DNA itself, because DNA is actually a digital information storage device. And it stores the information, the code script, in a form of letters, those base letters, A, G, C, T, that we've already discussed. And this is like listening to words, or it's like reading sentences, or, of course, computer bytes. But we like to think we invented all of this in recent history, but actually life invented it over 3 billion years ago. So we're just following up on what the humble bacteria-like organisms were doing 3 billion years ago, and understanding them is really best thought about in terms of information storage.
Now, to modify those genes, Mother Nature takes time, in fact, millions of years sometimes to make a key mutation in our genes. But today we have something called CRISPR technology. We had a guest a few months ago about CRISPR technology. And in some sense, doesn't that mean that we can short-circuit this process from millions of years to just a few days?
It absolutely can. I mean, we've been able to change in this precise way in some life forms, like yeast, for actually 40 years. So it's been around for some time. But the beauty of the new CRISPR-Cas discovery, and I know many of the people involved in it, is that you can use it with great efficiency on all living things, including ourselves. So we do have the ability to precisely modify our own genes, which could be used for medical purposes, for example, and certainly could be used in all sorts of different situations, model organisms, yeast, plants, mice, for example, to investigate how life works. So it's a really important technology that is going to be very, very important in the coming years.
Now, if we make a car that's defective, we could recall it. But a life form cannot be recalled. It reproduces out of control. Look at crops, mutant crops that proliferate in the environment, in a different environment, and we can't run the videotape backwards. Are you concerned at some point that there could be harmful accidents jumping out from the laboratory, and you cannot recall a life form? How are you going to put the genie back in the bottle if it's already out? What are your thoughts?
Well, I think this is something we really do have to think very carefully about, particularly with human beings, because modifying genes in human beings when you don't always know what the consequences are is really, in my view, an immoral thing to do. And there's some genes that have very simple outcomes. The cystic fibrosis is caused by a defect in a single gene, but many others are much more complicated. So you have to be very careful about this.
Thank you so much for being on Science Fantastic.
It's been a pleasure. Thank you.
Okay, once again, the book is called What is Life? 5 Great Ideas in Biology by Nobel laureate Paul Nurse.